Welcome once again to Bio 211, the Plant Physiology Unit 2, Session 2, Respiratory Substrates. And by the end of this session, students should be able to define respiratory substrate, name types of respiratory substrates, explain other uses of respiratory substrates, and state when they are used in the respiratory processes. Respiratory substrates are also referred to as respiratory substances. A respiratory substrate is any substance required for cellular respiration to derive energy through oxidation process. That is, it is that starting material which organisms need to carry out respiration on. The most common respiratory substrates are carbohydrates, lipids, and even proteins. The most common among them is carbohydrates, especially glucose. Though different organisms have evolved different pathways, usually respiration is carried out either aerobically or anaerobically using glucose as the respiratory substrate. So the more hydrogens a substance contains, the more ATP it produces in the electron transport chain. Some molecules have more hydrogens than others. The more hydrogen atoms there are in a respiratory substrate, the more ATP is produced. If there are more hydrogen atoms per mole, fixed amount of substrate, the more oxygen is needed to be the final acceptor. Usually, carbohydrates have fewer hydrogens than lipids. Similarly, lipids have fewer hydrogens than proteins. Therefore, more oxygen is required to break down protein than lipids and carbohydrates. A greater amount of oxygen is also required to break down lipids than carbohydrates. A carbohydrate is a biological molecule consisting of carbon, hydrogen, and oxygen atoms. Usually, the hydrogen-oxygen atom is 2:1, as in water. The empirical formula of carbohydrates is 2mH2ON, where M could be different from N. Some exceptions exist, as is found in the deoxyribose, a sugar component of DNA, which has the empirical formula C5H10O4. Here, we can see clearly that even though the hydrogen atoms are more than the oxygen atoms, the ratio H is to 0 is not 2:1. We can refer to carbohydrates as technically hydrates of carbon. Structurally, it is more accurate to view carbohydrates as polyhydroxyaldehydes and ketones, that is, aldehydes and ketones with many OH groups. The polyhydroxy nature can be observed in the straight chain structure of glucose shown below, as in glucose and fructose, an aldohexose and a ketohexose. So this is the structure of the aldohexose. Carbohydrates. The term is most common in biochemistry, where it is a synonym of saccharide. Saccharides include sugar, starch, and cellulose. The saccharides are divided into 4 chemical groups: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. In general, the monosaccharides and disaccharides, which are smaller, lower molecular weight carbohydrates, are commonly referred to as sugars. The word saccharide comes from the Greek word saccharon, meaning sugar. The scientific nomenclature of carbohydrates is complex. However, the names of the monosaccharide and disaccharide very often end in the suffix "-ose". For example, grape sugar is the monosaccharide glucose, cane sugar is the disaccharide sucrose, and milk sugar is the disaccharide lactose, as seen in the illustration. So monosaccharides are the simplest carbohydrates in that they cannot be hydrolyzed into smaller carbohydrates. They are aldehydes or ketones with 2 or more hydroxyl groups. 2 joined monosaccharides are called a disaccharide, and these are the simplest polysaccharides. Examples include sucrose and lactose. Sucrose is the most abundant disaccharide and the main form in which carbohydrates are transported in plants. It is composed of 1 glucose molecule and 1 fructose molecule. Do you know that sucrose is the common table sugar? The sugar you put in your tea, Milo, coffee, or cocoa is sucrose. Cellulose and hemicellulose are examples of oligosaccharides with just a few tens or hundreds of monosaccharides joined, while starch is a polysaccharide with thousands of monosaccharides joined together. Carbohydrates perform many roles in living organisms, including plants. Polysaccharides serve for the storage of energy, example starch and glycogen, and as structural components, example glucose, cellulose in plants, and chitin in arthropods. Saccharides and their derivatives include many other important biomolecules that play key roles in the immune system: fertilization, preventing parthenogenesis, blood clotting, and development. Glucose is the main respiratory substrate. Some mammalian cells, such as brain cells and erythrocytes, can only use glucose for respiration. In animals, excess glucose is converted into glycogen for storage, while in plants, starch is the storage product. Both glycogen and starch can be hydrolyzed to produce the monomer glucose whenever the body requires it. Other monosaccharides such as fructose or galactose may be converted to glucose to be used in respiration. The theoretical maximum energy yield for every mole of glucose is 2,870 kJ per mole, and it takes 0.6 kJ to produce 1 mole of ATP. So theoretically, the respiration of 1 mole of glucose should produce nearly 94 ATP. However, the actual yield is around 30 ATP, giving an efficiency of around 32%. The remaining energy is given off as heat, helping to maintain a suitable body temperature, allowing enzyme-controlled activity to proceed in animals particularly. Glucose is the most common substrate for respiration in most mammalian cells. Also, animals store glucose as glycogen and plants as starch. Theoretical maximum energy yield for 1 mole of glucose is 2,870 kJ. It takes 30.6 kJ to produce 1 mole ATP. Respiration of 1 mole glucose should produce nearly 94 mole ATP, but the actual yield is more like 30, as it has an efficiency of 32%. Remaining energy is used to generate heat. Lipids. You should know some lipids used daily as food or cosmetic. You might have seen shea butter, palm oil, palm kernel oil, fried oil, maize oil, canola oil, among others. Some of them are liquid, while others are solid at room temperature. These are lipids. Lipids are a group of naturally occurring molecules that include fats, waxes, sterols, fat-soluble vitamins such as vitamins A, D, E, and K, monoglycerides, diglycerides, triglycerides, phospholipids, and others. The main biological functions of lipids include storing energy, signaling, and acting as structural components of cell membranes. Lipids have applications in the cosmetic and food industries. Lipids may be broadly defined as hydrophobic, that's water repelling, or amphiphilic, both water-loving and fat-loving small molecules. The amphiphilic nature of some lipids allows them to form structures such as vesicles, multilamellar, unilamellar liposomes, lipid bodies with aqueous solutions inside, or membranes in an aqueous environment. Biological lipids originate entirely or in part from two distinct types of biochemical subunits or building blocks: ketoacyl and isoprene groups. Thus, lipids may be divided into 8 categories based on their origin. These are fatty acids, glycerolipids, glycerophospholipids, sphingolipids, saccharolipids, and polyketides derived from condensation of ketoacyl subunits, and sterol lipids and prenolipids, which are derived from condensation of isoprene subunits. The term lipid is sometimes used as a synonym for fats. However, fats are a subgroup of lipids called triglycerides. Lipids also encompass molecules such as fatty acids and their derivatives, including tri-di-monoglycerides and phospholipids, as well as other sterol-containing metabolites such as cholesterol. Lipids are a particularly important respiratory surface for muscle tissues. Triglycerides can be hydrolyzed by the enzyme lipase to fatty acids and glycerol. Glycerol can be converted to glucose and then used for respiration as normal. Fatty acids, however, cannot be converted to glucose. They are long-chain hydrocarbons with a carboxylic acid group at their terminus. This means they contain a great deal of carbon and even more hydrogen. The hydrogen atoms make them a great source of protons for the oxidative phosphorylation stage, as they produce a lot of ATP molecules. Each fatty acid is combined with coenzyme A, as shown in the diagram above. This reaction requires the energy of hydrolysis of one ATP molecule into a molecule of AMP and 2-phosphate. The fatty acid coenzyme A complex is then transported into the mitochondrial matrix, where it is broken down into two carbon acetal groups that are charged to coenzyme A. This breakdown process is referred to as beta oxidation pathway. During this breakdown, reduce nicotinamide adenine dinucleotide and reduce FAD are formed. The acetal groups are released and enter the Krebs cycle, where as normal they produce 3 reduced NAD and 1 reduced FAD and 1 ATP each. The large amounts of reduced NAD are reoxidized during the electron transport chain afterwards. During oxidative phosphorylation, very large numbers of ATP are produced due to the large number of protons available. If you do not understand this, it will be clear by the end of session 3 and 4. It is important to remember that lipids and proteins can only be respired aerobically and cannot undergo glycolysis. Both can be hydrolyzed into substances which enter Krebs cycle, and then the electron transport chain made of fatty acids and glycerol. So here is the fatty acid metabolism: the outer membrane and membrane, mitochondrial matrix, ketone bodies, oxidation in extrahepatic tissues. Then the process begins in the cytosol, where you have acetyl coenzyme A moving to acetyl carmethine, then acetyl carmethine coenzyme A moving in through beta oxidation and NAD and FAD, giving us NADH, FADH, and the process continues. So, summary of lipids: glycerol can be converted to glucose, but fatty acids cannot be converted to glucose. So, fatty acids contain many carbons and hydrogens. Fatty acids combine with coenzyme A after ATP has been hydrolyzed, split using water to AMP, adenosine monophosphate. So fatty acid coenzyme A complex is then taken into the matrix and broken down into 2 acetyl groups. Reduced NAD and FAD are formed. The acetyl groups are released from coenzyme A and enter Krebs cycle, producing 3 NADH, 1 FADH2 and 1 ATP. NADH plus FADH2 produced can then go to electron transport chain. So proteins are large biological molecules or biomolecules or macromolecules consisting of one or more long chains of amino acid residues. Proteins perform many kinds of functions within living organisms, such as catalyzing metabolic reactions, replicating DNA, responding to stimuli, and transporting molecules from one location to another. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes, and which usually results in folding of the protein into a specific 3-dimensional structure that determines its activity. A linear chain of amino acid residues is called a polypeptide. A protein contains at least one long polypeptide. Short polypeptides containing less than about 20 to 30 amino acid residues are rarely considered to be proteins and are commonly called peptides or sometimes oligopeptides. The individual amino acid residues are bonded together by peptide bonds and adjacent amino acid residues. Excess amino acids may be deaminated. Deamination involves removing the amine groups of amino acids and converting them to urea. This step is then followed by converting the rest of the molecule by glycogen or fat, which can be stored and respired later to release energy when required. When an organism is fasting or starving, proteins may be hydrolyzed to amino acids. which can then be respired. Some can be converted to pyruvate or pyruvic acid or acetic acid, which can be carried through to the Krebs cycle. NAD accepts a slightly higher number of hydrogen atoms during Krebs cycle from amino acids than it does from glucose. So respired amino acids have a higher energy yield, although only a small amount of amino acids may be involved. The mean energy value of carbohydrates is 15.8 kilojoule per gram. That for lipids is 39.4 kilojoule per gram and 17 kilojoule per gram for proteins. An organism will first use up its carbohydrate reserves before using up its lipid or fat reserve for respiration. After both carbohydrate and fat reserves are exhausted, then protein are used for respiration. So the integration of catabolic pathways is given. When a covalent bond is broken in a substrate molecule, sometimes that electron energy is transferred to a coenzyme and it is later used to make ATP when that coenzyme is reoxidized by the electron transport process. So since the concentration of coenzymes in a cell is limited, they must be reused. Thus, the reduced form must be reoxidized. Electron transport does that. The energy stored in is used to make ATP by oxidative phosphorylation. So excess amino acids are deaminated, removal of amine group converted to urea. Rest is changed to glycogen or fat, and protein is hydrolyzed, split with water to amino acids, which can be respired. So some can be converted to pyruvate or acetate and then is carried to the Krebs cycle. Some can enter Krebs cycle directly. The number of hydrogen atoms per mole accepted by NAD then used to in electron transport chain is slightly more than the number of hydrogen atoms per mole of glucose. So proteins release slightly more energy than equivalent masses of glucose. So in review, we defined and showed the types of substrates used in respiration. We also noted how and when the substrates are used. It is important to note that acetyl coenzyme A is the most important link in the whole process of respiration. It doesn't matter what the diet consisted of. It is the link that takes it to where all the important energy currency can be produced.